Today we're going to begin a discussion of systematics, which is the study of the diversity of life, both past and present, and the relationship of organisms through time. And as part of that study, we'll also be discussing taxonomy, which is the science of classification. Now, our approach to classification is going to be one of phylogenetics. And in phylogenetics, what we're trying to do is establish the evolutionary relationships of organisms via a process known as cladistics, but I'll get to that a little bit later. So again, our general approach to classification will be establishing evolutionary relationships as opposed to just lumping and sorting organisms according to similarities and differences. All right, so when we're classifying organisms and trying to establish evolutionary relationships, some of this we've talked about before, there are a number of factors that we can look at. So certainly one of those would be genetic analysis. And again, this has become a much more prominent way of classifying organisms. So we'll look at either DNA or RNA to try to look for a common ancestry of organisms. Because again, as I've established many times, shared genes means common ancestry. Additionally, we'll look, and so many of these other factors then are kind of indirect measures of genetic similarity. So for example, we'll look at morphology. Morphology is the study of the anatomy or shape of organisms. And so we can look at the fine anatomies, such as cells and tissues, that kind of thing. And also gross anatomy. So gross anatomy would be the morphology or the anatomy that we can see with the naked eye. So that would be organs, organ systems, bones, that kind of thing. Now, additionally, biochemistry can also be an important aspect of classification. So that could be the composition of organisms. What are they made of? It can also be the biochemical or physiological pathways, some of which we've talked about already. So for example, many organisms are heterotrophs, organisms that practice the cellular oxidation of glucose, something that we talked about in great detail, taking organic molecules, breaking them apart to generate ATP to carry out cellular processes. And then of course, many organisms are autotrophs. Autotrophs are so-called self-feeding organisms, and so photosynthesis would be an example of this. So autotrophs then are going to be taking inorganic molecules like carbon dioxide and water and combining those to form organic molecules, and in so doing, of course, that takes energy, and so they have to convert some source of energy to ATP and then use that ATP to build the organic molecules. So when we talk about autotrophs then, there are photoautotrophs, which are using light as that energy source for the production of organic molecules. And of course, photosynthetic organisms like plants would be an example of that. And then also there are chemoautotrophs, and chemoautotrophs are organisms that carry out inorganic chemical reactions that freeze up energy to generate ATP, and then that ATP can be used for the construction of organic molecules. So again, chemoautotrophs are different from heterotrophs in that they're using inorganic chemical reactions to generate essentially ATP. Okay, now again, continuing with other aspects of biochemistry that we may consider in classifying organisms, one would be the tolerance or toxicity of oxygen. So some organisms, like ourselves, are considered obligate aerobes. And so here, aerobe is a reference to oxygen. Obligate means that you have to do it or have to have it. So an obligate aerobe would be an organism that has to have oxygen to survive. And believe it or not, there are some organisms for which oxygen is actually toxic, and so they are obligate anaerobes. So anaerobe would be without oxygen. So they have to be without oxygen. So for example, bacteria in our intestinal tract, some of them are obligate anaerobes. There are a lot in the environment, microorganisms that are obligate anaerobes. And then there are some organisms that are facultative anaerobes, which means that they can tolerate oxygen or utilize oxygen as well as be in environments without oxygen. Okay, now still other factors we would consider in classification, embryonic development. Again, we talked a little bit about this when we discussed evolution. So again, we would look at similarities in embryonic development to suggest a common ancestry or relationships. And then also life cycles. And life cycles are a little more encompassing than just embryonic development. The life cycles would include such things such as modes of reproduction. It would include embryonic development that would also include metamorphoses or changes in anatomy that occur over development. Also, if it was a parasitic type of organism, it would include what hosts are being exploited. So life cycles basically include the entire aspect of reproduction for a particular type of organism. And again, that can give us clues to relationships of organisms. We also look at the ecological roles sometimes as clues to relationships of organisms. I would say this is probably of lesser importance than some that we've discussed. But for example, most organisms are quote unquote free living. In other words, they do not physically associate with other organisms. And other organisms do physically associate with organisms, and we refer to that as symbiosis. So physical association, again, for nutrition or shelter or whatever it happens to be between two different species, we define that as symbiosis. Typically, though, symbiosis refers to only eukaryotic organisms. I personally don't have a problem expanding that to both eukaryotic and prokaryotic organisms. But anyway, but again, the important part is the physical association between organisms. When we look at symbiotic relationships, there are basically three general types that we recognize. One type of symbiotic relationship is commensalism. In commensalism, one species benefits from the relationship and the other species is neutral in the relationship. So an example would be, for example, we have certain if we want to go into the prokaryotic world, we have certain microorganisms in our gut that really don't do us any harm, but it's a great place for them to live. So they benefit from that relationship, warm place to live, meals delivered, but they don't really hurt us. And another example would be, in most people, we have tiny arthropods called mites that live at the base of our eyelashes. And again, they can just feed on dead skin and secretions from the hair follicles there. And it's a good relationship for them. It's good for them, but we don't even know they're there. So they don't hurt us, they don't help us. That's an example of a commensalistic relationship. And those are called eye mites or eyelash mites. Mutualism occurs when both species in the relationship benefit. So an example of this would be in a lot of herbivorous animals. Animals cannot digest cellulose, and so there are microorganisms, unicellular organisms that live in the gut of termites, deer, cattle, goats, and so on, that can digest the cellulose. And so, for example, when a young horse or a baby cow is born, so a calf or a young horse are born, kind of instinctually what they'll do a lot of times is actually eat feces, or they may be licking the anus of the mother, for example. And so what they're actually doing is people look like, oh my God, how gross, but actually what they're doing is seeding their intestine with the microorganisms that they're going to need when they transition from feeding on mother's milk to feeding on grasses. And then the last kind of symbiotic relationship is parasitism, and this is where one species benefits and the other is harmed. And so parasitism then is the basis of disease. And when we look at parasites, we can classify them in one of two general categories. There are ectoparasites that we find on the outside of the body, so a tick or a mosquito would be an example of an ectoparasite, and endoparasites live inside of organisms, such as intestinal worms, for example, would be an example of an endoparasite. And hosts, we can also classify if you're and the host would be the one that's being harmed in the parasitic relationship. And a host would be an intermediate host if you are the host to a juvenile parasite, one that is not sexually mature. You're a definitive host if you are the host to a sexually mature parasite. And an accidental host would be one where you're not the normal host, but it actually parasitizes you nonetheless. And sometimes that's actually a very dangerous situation because usually host-parasite interactions are fairly old relationships. There's been a lot of coevolution ongoing there. And so a lot of times parasites have evolved to be less harmful. And so when you're not the normal host, a lot of times the parasite doesn't know exactly where it's supposed to go, what tissues to avoid to keep you healthy. And it ends up migrating to places it shouldn't go, may end up in brain tissue, may end up in spinal cord tissue, may end up in the eye, places where it doesn't really belong. So they can actually sometimes be more harmful than a quote-unquote normal host. Now, as we look at classification, historically we've used what's known as the Linnaean system of classification. And the father of classification was a Swede named Carl von Linné. And one of his legacies is that he's the one that Latinized sciences. He thought that science, European science, needed a common language, and so he decided to Latinize the classification scheme he came up with. And he was such a fanatic about this that he actually Latinized his own name to Carolus Linnaeus. So we refer to him now as Linnaeus, and his taxonomic scheme, the Linnaean system. And the Linnaean system is basically a kind of a nested box kind of approach, going from categories that are very mixed to ones that are very specific. And so in the Linnaean system, the largest grouping would be the kingdom. And kingdoms then could be broken up into smaller groups known as phyla or divisions. And the term phylum is used only with the animal kingdom now. Really, with all other kingdoms of organisms, the term division is used. And then divisions are still a pretty heterogeneous group or mixed group, and so divisions can be broken up into classes, classes into orders, orders into families, families into genera. Genera is the plural term for genus. And then each genus can be broken up into species, and of course we define species as like organisms, interbreeding organisms. And we've talked about that before. Now, the problem with the Linnaean system, however, is that it essentially dictates that there are a certain number of steps, basically, in evolutionary history, if you will. If we were comparing different types of organisms and their classification schemes, it's kind of saying, well, it takes this many steps to evolve to a species. And of course, as we look at relationships, many times there are additional steps. So the Linnaean system doesn't really fit a phylogenetic approach that well. And so we've had to add a number of different categories, and it's just grown beyond belief, to be honest with you. So for example, you might have superorders and suborders, or superfamilies, families, and subfamilies, and so on down the line. And just to give you an example of this, if we looked at this, and actually if we look at the Linnaean system for humans, let me do that first, we would be in the kingdom Metazoa, which are animals, phylum Chordata, animals with a spinal cord, class Mammalia, fur, mammary glands, primates, which would include monkeys, apes, and humans, among others, and also the prosimians. Hominidae would be the apes and humans, and then we're in the genus Homo, and the only living species of humans is Homo sapiens. So again, this would be how the Linnaean system to be applied to us. But as I was mentioning, we've added a number of super and subcategories, and just to actually show you another category that we've added, we've also added a category of domain. And so just to get from domain down to phylum, for example, you could go from domain to subdomain to superkingdom, kingdom, subkingdom, branch, infrakingdom, and others before you even get to phylum. So again, this Linnaean system has expanded tremendously, and sometimes we need additional categories to accurately reflect evolutionary relationships. Now, our goal in classification is to establish what we call monophyletic groups, or monophyly. And in that situation then, our taxonomic groups then all trace from a common ancestor. That would be the goal of monophyly. Organisms within a particular group to trace back to a particular ancestor. So let's say that we have a number of different species, A, B, C, D, E, F, G, and H. And again, remember, species are like organisms, and generally do not interbreed in our biological definition. And so let's say that species A and B trace back to organism I, type I, C and D, to J, E and F to K, G and H to L. So they have those as common ancestors. And I and J trace back to another common ancestor, M. And K and L trace back to N. And M and N trace back to O. So if we were looking then at taxonomic groups, we could identify A through H here as species. And we would lump then, or put A and B within genus I, because they both trace back to I. And we would put C and D within genus J, E and F within genus K, G and H within genus L. We could put all of these organisms within family M, because they all trace back to M. We could put all of these within family N, and they would all go in order O. So each of these then is defining a monophyletic group. Again, a monophyletic genera, monophyletic families, monophyletic order, and so on. Now, what we want to try to avoid, and one of the biggest sins, if you will, of classification would be what we call polyphily. And polyphily is where you include organisms that don't belong in your group, organisms that do not immediately translate back or transfer back to a recent common ancestor. So for example, again, we could have these two monophyletic groups here, and then if we go ahead and if we look at, so again, these would be monophyletic groups. So let's say we have a genus K, that includes species E, F, and G. This would be an example of polyphily, or a polyphyletic group. So we've included G in this particular genus, even though it does not trace back to the common ancestor K. So that would be a problem in classification. We wouldn't consider that a monophyletic group. That's a polyphyletic group. And then again, I'll come back to this in a second, but then we now would have a group then that would be incomplete, and we call an incomplete group paraphyletic group. So these are both problems. We don't want to have polyphyletic groups, and we don't want to have paraphyletic groups when we can avoid them. Sometimes it's unavoidable, but we try to avoid that. So again, this would be another example of polyphyly or polyphyletic group where we now have a family with a genus K that we've included that doesn't belong here. And again, this would be a paraphyletic group that's incomplete. So again, paraphily then would be incomplete groups. Again, if we don't trace back to a complete group, so again, if we had an order here that is including some that shouldn't be included, that would be polyphyletic, and then that would leave a paraphyletic group. Okay, now, our approach to classification is also significant. So for example, this is a picture of my daughters. This is Valerie and Melanie. And they look quite a bit alike in this particular picture, but Valerie is very light complected. She actually used to have blonde hair when she was younger. She's blue-eyed. And Melanie is really very dark complected, dark hair, dark eyes, and looks a lot more like her mother, to be honest with you. But what's interesting too is that Melanie also looks a lot like her cousin, Chris, and I don't have a picture of Chris, but Chris is also dark complected, dark eyes, and they actually look very, very much alike. They look like they are more closely related to one another than Melanie is to Valerie. So again, if we were putting together a family tree, if we based that family tree just on appearances, then Melanie and Christopher actually look more alike than Melanie does to Valerie. And so if you were putting together a family tree just based on appearances, you would end up linking Melanie and Christopher together rather than Melanie to Valerie. So this would be one approach. But this is not an approach that would show you the relationships of the organisms. This would just be lumping according to similarities and separating according to differences. Now, if you did make a family tree based on relationships, then, of course, Melanie and Valerie would have a more recent common ancestor than either would have to Chris. So this would be the appropriate family tree. So again, we use the same kind of approaches then when we look at evolutionary relationships as well. So in a so-called traditional approach, kind of the Linnaean approach, if you will, basically, organisms are grouped according to similarities and separated according to differences. And so let's take three organisms, trout, lungfish, and cow. And let's say that we want to classify these, generate a family tree. Now, this seems pretty easy. I've got a fish, and I've got a cow. So obviously, you'd put the fish together. They should be more closely related, and that would generate a family tree that looks like this. And again, that's very intuitive. It makes a lot of sense. And how could you be wrong with that? Well, that's one approach. Now, there is a different approach that's really better at establishing evolutionary relationships. And we refer to this as a cladistic approach. And in the cladistic approach, what we try to do is identify what we call ancestral characteristics, more ancient characteristics that have evolved earlier that we see in the fossil record, and derived characteristics. And derived characteristics are more recently evolved characteristics. And the concept here is that if organisms share recently evolved characteristics, then they have to have a more recent common ancestor. So in a cladistic approach, we're not necessarily interested in all characteristics. We're interested in the most recently evolved characteristics. And those that share more recently evolved characteristics have a more recent common ancestor and are therefore more closely related. And this can be very counterintuitive. So if we go back to our example of trout, lungfish, and cow, again, intuitively, you would put trout and lungfish together. Fish or fish, put fish together. But if we look at this from a cladistic approach, the lungfish does have a lung-like organ. And let's say then that a gene has been identified that is responsible for the lung of the lungfish, and it also triggers development of lung in a cow. So that's a recently evolved gene that's responsible for lungs. Gills are the ancestral characteristic then. So if lungfish and cow share a gene, which is a more recently evolved gene than the gene for gills, then what that translates to in a family tree is that we would actually have the lungfish and the cow linked together in our family tree. So this is counterintuitive at first, but it's really not that much different than the family tree that we talked about with Valerie and Melanie and Chris. So when we build trees based on appearance, we're hoping that things that look alike have a more recent common ancestor, and many times they do. A lot of times that works out. But in the cladistic approach, it's a more refined approach, so we're more interested in recently evolved characteristics, meaning, again, if you share recently evolved characteristics, you have to have a recent common ancestor. So this, believe it or not, is the more appropriate family tree than the previous family tree. So the cladistic approach is better from a phylogenetic perspective, one where we're trying to establish evolutionary relationships and not just organize for the sake of organizing. Again, another approach too, we could look at a gorilla, fossils from Australopithecus and Homo. And so when we look at Australopithecus and Homo, we see that they share a number of derived characteristics. Tibia for walking, the angle of the femur for walking, all of these characteristics for walking are recently evolved. And so again, that's why we link Australopithecus to humans in a family tree, more so than to say a gorilla or a chimpanzee or something like that. Okay, now, when we look at the organization of life then, in addition to the kingdom level that Linnaean had, the modern approach is to add another level of classification here, which is the domain level. And we recognize 3 domains of life, the eubacteria, the archaea, and the eukarya. And so when we look at the the eubacteria and the archaea, what they have in common is that they are prokaryotic organisms. And so you may recall that prokaryotes are small, they're simple, they don't have a true nucleus, they don't have a lot of membranous organelles, they have circular chromosome, characteristics like that. And the eukarya is composed of all the eukaryotic organisms. And again, we talked about that in earlier modules, but again, large cells, true nucleus, membrane-bound organelles, and so on. And we compared animal and plant cells at that time, but there are other types of organisms as well that are eukaryotes. Now, in looking at the relationship of these groups and general characteristics of the group then, first, what makes bacteria bacteria? What characteristics are unique to bacteria? What I would like you to know about the bacteria is that, and again, there's a lot of technical terms here on this chart, but I want to kind of simplify this for you. What I want you to do in terms of defining bacteria, what makes bacteria bacteria is that they have unique cell walls. They have something called peptidoglycans in the cell walls. So that's what's unique to the bacteria. Now, the archaea, what characteristics are unique to the archaea? Primarily that they have very unusual plasma membranes. So they have, and again, we don't need the details here, but I'll get to that in a second, but they have very unique phospholipids in their plasma membranes. So in their plasma membranes, they have phospholipids that will actually go all the way across the membrane, at least some of them, and also their phospholipids have branched fatty acids. And so that's unlike any other organism, and that's what makes the archaeans the archaeans. Additionally, as we look at the eukarya then, what makes the eukaryans eukaryans? They're eukaryotic, to be quite honest about it. So again, they have a membrane-bound nucleus, membrane-bound organelles. So now as we begin to look at relationships that are shared by the groups then, and what's the relationship of our 3 groups? And again, eukarya are eukaryotes, that's kind of the bottom line there. But if we look at what are shared characteristics, the bacteria and the archaea then have basically prokaryotic characteristics. So they lack a nuclear envelope, they lack membrane-bound organelles, they have a circular chromosome. So basically all of the characteristics that are prokaryotic characteristics, they have those in common with one another. Now, as we look at the archaeans and the eukarya, and we look at what characteristics they have in common, what we find is that they lack peptidoglycans in the cell wall, for example. And then a number of characteristics, again, that we would consider derived characteristics. For example, their messenger RNAs have a particular signature. For example, their messenger RNAs only code for one protein instead of multiple proteins, for example. They always begin with methionine in terms of their protein production, and some other characteristics as well. But the particulars here aren't as important as what I would like you to know is that these characteristics are considered derived characteristics. So when we look at the archaea and the eubacteria then, or excuse me, the archaea and the eukarya, I have to be careful here, they share derived characteristics. So as we look at characteristics of the 3 groups then and try to decide what's the relationship of these 3 groups then, originally it was believed that the archaeans were the most ancient group, and that's why they're named the archaea. And it was thought that eubacteria perhaps evolved later and eukarya after that. But because the archaea and the eukarya share what we consider to be derived characteristics then, as we put together a family tree, these are the 2 that we would end up lumping together. So again, this is somewhat counterintuitive because I think intuitively people want to lump prokaryotes together. And historically that's what we did. But again, when we look at the actual characteristics, those that we consider more recently evolved show a more recent common ancestry between these 2. So again, the archaea are somewhat inappropriately named. Now, when we look at the three domains then, we're going to be getting into prokaryotic classification really more so in our next unit. But one thing I do want to mention is that what's really important characteristic or really important discovery in helping us identify and characterize prokaryotes was actually the development and discovery of a substance called agar. And so agar is actually derived from red algae, and it's a really important substance because it cannot be digested by bacteria. And so I'm going to show you on the overhead here what agar looks like. And so anyway, we use these agar plates, and again, the composition of agar is it's kind of a gelatinous material. It's soft and so on. But the real importance of agar is that, as I mentioned, is that it's a substance that bacteria and other prokaryotes cannot digest. And this may not seem like a big discovery, but this is a really big deal because bacteria cannot digest the agar, as bacteria grow on that agar surface, then they stay separated from one another. And so this allows us then to grow what we call colonies of bacteria. And I'll come back to this in a future module, but these are kind of light. But you can see on this agar plate that we have colonies of bacteria that are growing, but they're staying separated. And you'll see that a little bit better here because these are a pigmented type of bacterium. So we can see the individual colonies here. So what that allows us to do, or really where each of those colonies come from actually, is from an individual bacterium. So what we start with in these plates is we'll actually streak out a mass of bacteria and then streak them out across the plate through various techniques and end up with individual bacteria on the plate. Then they will divide, and so each colony then is derived from a single bacterium. So then we can further grow those bacteria and learn about their characteristics, or archaeans would be the same kind of technique. So the importance here is that because agar cannot be digested, what used to happen with gelatins like Jello-type gelatin is that the bacteria would begin to grow. They would digest the gel, and then they would end up liquefying and flowing together. And so it was impossible to get individual bacteria isolated from one another. So we'll talk more about prokaryotic characteristics, but understand that one of the really important fundamental discoveries was agar, and that allowed us to isolate individual bacteria and archaeans, and then develop procedures to learn about their individual characteristics. So that's it for now. I'll see you next time.